Good afternoon and welcome to today's webinar, Genetic Factors in Autism, brought to you by the University of Iowa Division of Child and Community Health, Child Health Specialty Clinics, and the Regional Autism Assistance Program. I'm Sean Wittrock and I will be moderating today's session. The session will be recorded and the link to that recording will be provided in an email following the webinar. You are free to type your questions into the Q&A box throughout the presentation, and those will be answered at the end of the presentation. Continuing education credits will be available for those attending this live session. Directions for obtaining those credits will be shared at the conclusion of today's session. I am pleased to introduce and welcome our presenter today, Dr. Jake Michaelson. Dr. Jake Michaelson is a Roy J. Carver Professor of Psychiatry and Neuroscience and the Division Director of Computational and Molecular Psychiatry at the University of Iowa. His lab studies the effect of genetic variations on the development of the brain, with specific applications in autism and language impairment. He earned his BS and MS in biological engineering at Utah State University before earning his PhD in computational biology at the University of Dresden in Germany in 2010. After his time in Germany, he joined the lab of psychiatric geneticist Jonathan Sabat at UC San Diego, where he completed his postdoctoral training and published several of the earliest papers dealing with whole genome sequencing in autism. In 2013, he joined the faculty at the University of Iowa, and his research is supported by NIMH, NHGRI, NIDCD, and the Simons Foundation. Research areas include genetics and comorbidities of autism, genetics of language and impairment, gender diversity, predictions of top autism gene candidates, and genetics of twice exceptionality. So without further delay, I will turn the presentation over to Dr. Michaelson.
Thank you, Sean, and thanks for the invitation to come and talk a little bit about the research that we've been doing. I'm sure that some of you have probably seen me speak in other venues, and I apologize if some of the content is duplicated. I also apologize, I'm on the tail end of a cold, feeling a lot better now, but it still is going to affect my voice and presentation a little bit. So here's the disclosure slide that I have to put in. There's nothing to disclose here. And I'm going to split today's discussion. Actually, I should say I made a few last minute changes in response to some of the pre-questions that were submitted. Just in the moments before we started, Sean shared a few of those with me, and I stuffed a few slides from another talk to hopefully address some of those. And I hope to leave plenty of time for discussion. I know Zoom webinars are not the greatest format for discussion. I wish we could be together in person and talk about these things, but hopefully we'll be able to have a discussion. Anyway, the first part, I'm going to give a general overview of how we do genetic research in autism and why it's important. And then in the middle, in between these two parts, I'm going to talk a little bit more about some of the questions. In particular, one question was raised about, with where we are right now with genetics, is there anything that could conceivably change about the clinical care of an individual with autism if we knew something about their genetics? So I'm going to talk a little bit about that in the middle. And then with whatever time remains, we'll talk about some of our more recent research on autism and suicidality, which I think is really important for clinical providers to have at the forefront of their concerns when they're helping autistic young people. Okay, so we're not going to go over all these questions today, but I like to frame things whenever I give a talk in terms of thought-provoking questions. And so, like I said, we're not going to talk about each of these, but I hope that they can stimulate some thinking that moves beyond just the nuts and bolts that we usually think about when we talk about autism. But first of all, what is autism? Is it a disorder or is it a difference? What's the opposite of autism? And what effect has the clinical definition of autism and the fluctuation of that clinical definition had on the community? What are the benefits of participating in research? What do scientists get out of studying autism? And then you find out that gene X, whatever that might be, is a major causal factor in your autism. Well, so what? And what does it mean if you're diagnosed with X, but your innate risk indicates Y? Okay, so here I just want to tell a little bit about Spark. I know many of you probably know about Spark from some of the work that we've done as one of the recruitment centers for it. But Spark is the largest ever study of autism, and this is a now very outdated map. But even back when it was created, you could see that basically wherever people were living in the United States, there were Spark participants. There's now over 300,000 participants in this study, and many of them with genetic data associated with the other things that they tell us about their autistic experience. And one of the ways that I think about one of the motivations for this kind of very large scale research of autism is that we're trying to make a map of autism, because I think anybody who has a lived experience with autism, or who has a loved one in their life who has autism, you understand that there's a great deal of diversity in the expression and the manifestation of autism. And so, in some ways, we're kind of on an expedition. I so over spring break, I was in St. Louis and went to the Arch. And if you've been there, you've seen that they have a lot of exhibits that touch on westward expansion and Lewis and Clark and so forth. And so this is kind of still on my mind right now, but there are a lot of unknowns, sort of like there were many unknowns for Lewis and Clark, and their directive was basically to just describe and to experience and to observe. And that's kind of what we're doing with Spark with autism now. Now, obviously, the hope, the motivation behind all of that is so that we can use that knowledge that we collect and that we synthesize to help autistic people reach their potential as a fully realized human being, who um, that they're able to capitalize on opportunities, that they're able to take advantage of their strengths, that they're able to um, uh, transcend any disabling aspects of autism that they might face, and live a fulfilled and a fulfilled life. Um, so there, yeah, that speaks to kind of the first point. The second point is that we want to improve our biological understanding of autism and how the brain works. Um, and so the Spark is definitely a study that's rooted in biology. There are lots of different ways and different lenses through which you can study autism. But this is a very it's kind of a hard science approach to understanding the many forms and varieties of autism. And I'll just point out the URL that you can go to if you're interested in learning more about Spark. I'm going to give kind of a lightning tour in the next couple slides. But you can join online. You don't have to go in anywhere to enroll. You can do it all in your home. If you do that, if you sign up, then you'd create an account online, you invite family members, and then you would get a box in the mail, and you would there's a tube in there that you and your family members would spit into, and then that would be returned. That would get sequenced at one of our lab facilities, and then that's just entering through the front door. And then as long as you're in Spark, you'll be given opportunities to participate in many different kinds of research. We've been very active in doing a lot of follow-up studies with Spark participants. And so I would say that the landscape is changing in a way in autism research that if you want to be involved in autism research, either as a researcher or as a participant, the most efficient way to do that is to join Spark, because it's not just one study. It's sort of the gateway into being in lots of other studies, because that huge community that Spark has, researchers go to Spark to answer questions about autism. And so if you want your experience to be represented in what scientists and doctors know about autism, then it makes sense to join this community. So you get some initial compensation. There's additional compensation available for the subsequent studies that you might choose to participate in. And then there's a lot of resources that are science-based, very rigorous resources. There's also the opportunity if there is a genetic finding associated with your sample that you turn in, then you would get that returned to you at no cost with genetic counseling. Now, you can either opt to have that returned to a provider of your choice, and then the obligation to interpret and explain the results would be on them, or you can have that returned to an online genetic counseling service. They're trained in genetic counseling, and they would be able to consult with you on the genetic finding. So this is a very conservative list. Obviously, to meet clinical criteria, we're very careful and we're very conservative. And so because of that, results are only returned to about 10 or 15% of participants. We could be much more relaxed and much more liberal than that, but there would also be more noise in the results that we would return. Okay, so a little bit about what we know about the genetic basis of autism right now. So first of all, I'm going to talk the talk today is going to be varying degrees of technical and hopefully other parts that are more lucid and more approachable. I'll try to emphasize everywhere where there's a place that I think is a key take-home message, and this is one of those. So there have been several studies over the past decade now, involving millions of individual data points of individuals. And they've all pegged the heritability of autism in the range of 80 to 85%. Heritability is a measure that scientists use to quantify the level of genetic influence in a trait. So it's between 0 and 1, or 0 and 100%, depending on whether you're measuring it as a percentage. So this is very high. Other neuropsychiatric conditions like major depression is more in the range, depending on how it's defined, probably in the range of 60%, 50, 60%, something like that. Schizophrenia would be in the 70s, maybe up to 80 or so. But autism is extremely high among neuropsychiatric conditions, very, very genetic. It's really among the most heritable traits that we know about. So this is important because for a variety of reasons, but one of the things that I say up front, this is one reason why there's been no data to support the popular myth that autism is caused by vaccines, or if not vaccines, some other kind of circulating myth of X, Y, or Z causes autism. With such a substantial proportion of the risk mechanisms of autism being genetic, there's very little room left over for other non-genetic contributing factors. Now, this isn't at 100%. There are certainly things that can, environmental factors that have been studied that can tweak risk a little bit in one direction or another. So, for example, air pollution has been one that's been shown to slightly increase risk. But they're all quite minor compared to the aggregate effect of genetics. So the interesting thing about these studies is that even though millions of people were involved, in these big heritability studies, actually no DNA was examined in any of these. So we can determine that something is strongly genetic just by looking at family relationships and recurrence of certain conditions. And that's essentially what was done here. So we can know something's very strongly heritable and still not fully understand all of the genes that are involved and the processes that are involved. So that's another thing that I think is important to emphasize, that although we have very compelling evidence that autism is strongly genetic, what we're still working on right now is understanding what genes are involved, what processes are involved. So just a quick primer to hopefully your high school / college level genetics. Just a reminder that we each have 2 copies of every gene, or you can think of it as 2 copies of your genome, one from your mom and one from your dad. And if you look at any given position in the genome, so here we're looking at this position, and on one copy, you might have a G at that position, and in another copy, you might have an A. And that can kind of separate it into these different states that we call genotypes. So if you look at a particular place in the genome or a particular gene, you have these gene variants. And we say that if you have two Gs, you have the homozygous alternate. Heterozygous is if you have one of each, and then homozygous reference uh, is if you have the two As. So the reference is just referring to the referenced human genome that we have, whatever is listed there as the DNA base. So to think about how these differences in gene variants can have an impact on traits that we're interested in. So here this is a language score that we would be working with. And you can imagine that if we looked at a particular place in the genome and just asked of all the people who are in our study, and they have these different combinations of gene variants and genotypes, if we group them by what their DNA says at this particular place in the genome, do we see any group differences in the trait that we're interested in? Well, here in this example, you can see that there's no differences between these groups. I might look at a totally different place in the genome, and if we were to group people by what gene variants they have there, we might see that there's a big difference. So in this case, people with an AA have significantly lower language scores, whereas people with AG are kind of in the middle, and people with GG are at the higher end. So this is a very kind of simple explanation of what we're doing when we kind of march through the genome looking for places where group differences would explain differences in a trait that we're interested in. And so if you do this at every position in the genome, you end up with a plot that looks like this, which geneticists call a Manhattan plot because it sort of resembles the skyline of a big city like New York, where you have very few skyscrapers kind of poking up above the horizon there. And then this is the statistical strength of the association represented with the negative log 10 p-value of that. And so basically what that means is if you look at these places where there's no peak, then what that means is that there's no relationship between the gene variant that you have in that place in the genome and the trait. And then wherever there's a peak, that basically means, yes, there is a group level difference where people can kind of be separated by their genotype, and it lines up with whatever trait you're interested in. A cool thing that we can do, which I'll talk a little bit about today, very little bit about, but I think is interesting to know, is that we can take all of the data from these, we call them GWASs, or genome-wide association studies. We can take the data that comes from those studies, and we can use them in we can take basically a summation of all of the effects across your entire genome, and then we can calculate a score that represents your propensity for the trait that was studied in the GWAS. So if it was language here, language score, like I was saying before, we could calculate your overall genetic propensity to have a high or a low score on that language test. And this has applications in research. It has applications in personalized medicine, because effectively what this means is that we can quantify in a single number your overall genetic propensity for a given trait. And we've done a lot of work with these polygenic scores. You can calculate a polygenic score for almost anything. So I'm going to talk a little bit about common genetic variation and then rare genetic variation. So common genetic variation just means that these are variants that are seen in a lot of people. So anything greater than 5% frequency is something that we would consider common. These changes, genetic changes, have very weak effect sizes, but in aggregate, they can have a profound effect. And we'll talk about that in just a minute. But these genetic variants have been around for many, 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 many generations, and they have been subjected to many rounds of selection, which means that because so many human beings carry these common variants, they by themselves, an individual common variant has very weak risk for disease, for instance. But their cumulative effect can be very informative, like in the polygenic scores that I was just talking about. Now, rare genetic variation. So everybody has rare variants too, and there are many more of them than common variants. But these variants arose much more recently in your family's history, and they've undergone fewer rounds of selection. And the distribution of effect sizes for any given rare variant is much broader than the common variants, which are mostly weak. So rare variants can be, totally harmless, or they can have a very big impact on your health. And these rare variants include what we call de novo mutations or new mutations. These are new mutations in a child that did not exist in either parent. And just to note, if you're referred for clinical genetic testing, typically they'll be looking for rare variants. I should emphasize every single person on this call who's listening to this talk, we all have de novo mutations. It's a totally normal part of reproduction. And in fact, it's good for evolution because it provides some diversity for evolution to act on. And we have between 70 to 100 single letter changes in our genomes that make us completely different from either of our parents. And depending on where those changes land, if they land in genes that are very important for brain development, then that can have a big effect on our development. It can have a big effect on autism risk or on ADHD or on many other neurodevelopmental conditions. So again, here's a list of autism risk genes. This might look like a long list, but you have to consider that the human genome has about 20 to 25,000 genes. And we think that the real list, the more comprehensive list of genes that contribute to autism risk could be 3 to 5 times longer than this list. Some of the things that we've learned about just really quickly about what constitutes an autism gene is that these are genes that tend to be expressed in the brain, because not all genes are expressed in every tissue in your body. There are many genes that are very specific to being used in a particular tissue. These are genes that are generally expressed during fetal development. They're kind of boss genes. They regulate the expression of other genes, so they're kind of master regulators. Many autism genes are also involved in neuronal synapse structure and function. And so one hypothesis that we have when we look at these different autism genes is that this is partly contributing to the major amount of diversity that we see in the presentation of autism and the experience of autism, because the flavor of autism that a person presents with depends on the combination of genes that are affected in that individual. So just to give you an example, and this is a slightly more technical thing, but a few years back, we published a paper in Nature Genetics with other Spark researchers. And in that, we found that there are 6 archetypal risk mechanisms for autism. So each point that you see on this graphic represents a different gene. And they're sort of positioned in a space that is related to their function in our biology. And so these, I won't go over all of these because I expect these are probably don't mean a lot to you, but neurotransmission, that first one up there, well, maybe I'll talk about the first two. Neurotransmission is related to all of the proteins that are needed for neurons to fire and to communicate with each other in the way that is needed for an organism. And then chromatin modification is sort of one of those regulatory protein or processes that I was telling you about. One thing that we've seen, interestingly, is that autistic people who have more than expected genetic burden in these neurotransmission genes, they're more likely to have seizures as part of the overall presentation of their phenotype. And so that's a way that biology kind of connects to the clinical presentation. And another example is that chromatin modifiers are they're very important and in regulating kind of system development and morphology. So people with a disproportionate number of rare variants and mutations in these genes will tend to have more dysmorphia and more characteristic, for instance, facial features and growth phenotypes and things of that sort. And so that's another way that we can kind of connect the clinical presentation with the underlying biology that's going on under the hood. Now, I talked about a couple of different modes of genetic risk, and thing that I think is important to understand is that there's no single mode that's operating in an individual. They're all operating in all of us at the same time. So we have each of us will have a couple of uh, you know big impact rare variants. We'll have many more moderately penetrant variants, and then we have kind of a sprinkling of a lot of different these common variants that have weak effects, and then together that kind of makes up a composite risk for a given condition. Okay. Um, this is a graphic from, I don't know why the resolution's coming through so poorly on this. I apologize for that. It shouldn't look like that. But this is from a paper that we wrote a few years ago that was a molecular case study, and I think illustrates what is happening with these different kinds of risk. So let me just walk you through this here. We did whole genome sequencing on a family with twin boys with autism. And we wanted to understand how the one mode of genetic risk, which was rare variant burden, and another mode, which is polygenic risk, these many common variants summed up together, how they kind of interacted with each other to underlie autism. And so this pink distribution, this pink blob is a reference distribution from a project called the 1,000 Genomes. These are people who don't have any kind of a neuropsychiatric diagnosis or history. And so we took their genomes and we calculated their rare variant burden in autism genes as well as their polygenic risk for autism. And then so that when you do that over enough people, that gives you this expectation, this distribution for a person who does not have a diagnosis, where on average their joint risk is. Okay, and you can see it's centered over here at 0, essentially. And we then projected the family on top of this to kind of understand what was going on in terms of genetic risk for the family. And the father had, slightly elevated rare variant burden, but was really average in the polygenic risk. But he was still very much in the territory of the expectation for a person who's not diagnosed. The mother had elevated both rare variant risk and polygenic risk, and was kind of on the outskirts of the expectation for an undiagnosed person. And then the twin boys who were diagnosed with autism were further away still from the expectation for an undiagnosed person, meaning that they had high levels of inherited rare variant burden, but also polygenic risk. I think the thing that's interesting to pause here and note, because I think this might be counterintuitive for some people, is how can a child um, end up being more extreme in their risk that they inherited because they got it from both parents, right? Why would they be more extreme? Well, it's like shuffling a deck of cards. Every time a child is conceived, there's a shuffling of the deck of the risk and protective variants that each parent has, and you'll get a distribution that is basically centered at the mean value for the parents. But there's tails of that distribution that extend in either direction, such that you can, it's possible, and it happens all the time, that there are children who inherit more genetic risk than either of their parents has on their own, and we think that's probably what's happening here. The final thing that I'll say, and I put this in almost every one of my talks because I think there's so much to talk about right here. And this is one of those moments where I wish we could be in the room together, because I think there's a lot of interaction, a lot of back and forth that can happen. But I want you to take a moment and pay attention to these margins here on the side of the graph, both here and here. And I'll just draw your attention to the lilac shade or the lavender shade is female. The green shade is male. And before I launch into what this means, I just want you to kind of notice that these distributions, and they're referring to our kind of our background distribution of people who don't have a diagnosis. But the male distribution is shifted lower here, and it's shifted lower there with respect to females. So think about for a moment what that means. That means to be in a group of people who are undiagnosed, a male can tolerate less genetic risk than a female can and still be undiagnosed. This speaks to what is called in this line of research as the female protective effect. We know from animals, well, we know from a lot of different sources of information, but in general, male development, male neurodevelopment across mammals at least, and even many other organisms, male development is much more fragile than female development. Female development tends to be more robust against genetic and environmental insult compared to male neurodevelopment. And we think this is one manifestation of this. Now, there's another separate discussion that we could have that I think is very interesting, because there's a lot of talk today about misdiagnosis and misdiagnosis of females with autism. We have a book club in our lab, and we've been reading Unmasking Autism, which I'm sure is a book that some of you might be familiar with. And there's a lot of discussion about why some people are not diagnosed. So I think that that is an issue, and that there is a lot to be said there. I will just say that that even if that's accounted for, even if those diagnostic biases that do exist, if they were completely removed, we would still see an excess of males diagnosed with autism. And it's because of very core biological developmental reasons. They're not cultural. There's definitely cultural factors that influence what the diagnostic criteria are and how they're applied. But even extracting that, and when you look in animal systems that don't have those layers of culture and bias that humans do, you see the same things. Males are more neurodevelopmentally fragile than females are. Okay, so that's all I'll say about that, but I hope that in the discussion time we can maybe talk a little bit more about that. So here's some take-home messages from this part of the talk. First, just remember that autism is highly genetic. It's in the 80s percentage-wise for heritability. There are very few cases of autism that can be completely attributed to a single gene. There's about 100 to 200 currently known autism genes, that may be up to 1,000, depending on how we define what contributes to autism risk. Females can tolerate a higher burden of risk factors than males and remain undiagnosed. So that feeds into the female protective effect. There's both inherited and de novo risk. So most of the risk that you're going to inherit is or that you have is coming from your parents. But there are de novo factors that all of us have. But if you have a de novo mutation that happens to impact a gene that is critical for neurodevelopment, then you're much more likely to present with autism. And right now, we can't predict who will develop autism based only on their genome. That's just not something that in our current state we're able to do. We can definitely assign probabilities, and we can definitely assign odds ratios and things and expectations, but we can't predict with certainty people who will develop autism. I think that's important. I say that, and I'm not drawing a straight line here, but there's often a lot of concern about from some parts of the autistic community that people should be worried about genetic research because it's ultimately leading towards eugenics for autistic people, which I'm here to tell you is not the case. It simply is not. I understand the concern, I understand the history, and I understand where that concern comes from, but that's not the case. And a big part of that is because autism is so complex, it's not possible to predict it just based on somebody's genome. Okay, so here's where I injected a few slides to hopefully be responsive to some of the questions about, genetics. So what? and I hope we can have a discussion about this. So this might be a little bit jarring because these are taken out of context from a different presentation. But I did want to start with, in talking about this, with thinking about the genetics of height, because height is like the poster child for a successful genetic dissection of how the genome influences height. It's a very simple trait, very straightforward to measure. It doesn't have nearly the uh, difficulty in defining that autism does. But a year or two ago, there was a genome-wide association study on height that reached what they called saturation. And basically what that meant is they had recruited so many people, or they had so many people in this study that adding more people was not going to discover new places in the genome that were associated with height. So that's what saturation meant. That took 5.4 million participants for a trait that is as simple as height. So you can imagine, you kind of do the mental math of like, if we're talking about something as complex as autism and as difficult to define as autism, you know, tens of millions, hundreds of millions, billions of people would be needed to reach saturation. So in that, they found 12,000 independent places in the genome that are associated with an effect on height. 12,000. And they explained 45% of the variation in height. And I'll just tell you from personal experience, I have my genetic data. And so I took the data from this and plugged it in. And the polygenic prediction for my height is accurate to less than an inch of error for me. So that's the level of predictive ability that is possible with if you reach saturation. And so I think it's this isn't talking about autism specifically, but just psychiatry. I think genetics has made an impact on how we are developing treatments in psychiatry. So the latest genome-wide association study for schizophrenia was in 2022. They had 75,000 cases and 100,000 controls. They found 287 distinct places in the genome that were associated with schizophrenia risk. This paper, even though it came out just a couple of years ago, has already been cited by 246 other papers, and many of them are pursuing mechanistic experimental studies of candidate genes that came out of this study to think about new therapies for schizophrenia. And so the other thing I'll say is that this GWAS was the main driving force behind the immune hypothesis of schizophrenia, which is related to synaptic pruning by microglia. And there have been at least 10 clinical trials spawned by this lead alone. So yes, genetics can provide key biological insight into what is actually happening in the brain. That can have therapeutic implications. So you can include genetics in your practice today. So I know not all of you are at the University of Iowa, but Ben Darbrow, pictured here, is the director of the cytogenetics lab. And I just want you to know that clinical microarrays, which is a way to do clinical genetic testing, are paid for by most insurance providers. And so if you are a provider, you have the ability to order this. And it's indeed recommended by the ACMG if you're working with an individual diagnosed with autism or with intellectual disability or with another developmental disorder. It is recommended to order genetic testing. And so that can provide insight into the causal mechanisms that can help a person develop a more specific identity of what they are working with and strengths and disabilities. It can connect you to other people, to communities, to online communities who have the same genetic risk factor. There are many clinical trials these days where they're looking for people with a specific genetic variation. And it can open doors for research too. So I think those are just some of the reasons why you should consider encouraging your clients to seek genetic testing. So it should be ordered in every case of a developmental disability, which includes ID, autism, language disorder, ADHD. And Ben and I have been continuing to look into adult psychiatric conditions and the feasibility of getting those covered by insurance. So again, this is a little bit out of place here from a different talk, but I just want to say that psychiatric traits in general are highly heritable. And psychiatric genetics hasn't even yet gotten close to reaching saturation, but it's already changing our understanding of the mechanisms, and it's providing new insight for therapeutic leads. Okay, and to give just a couple of anecdotes too about how, because I think the question that was asked that Sean gave me was, is there any, can genetics ever have any hope of actually changing anything that we would do clinically? So this is from a paper that we published a few years ago where we took drugs that were already FDA approved, and we just scanned through them to see if we could find evidence that some of these drugs that you might not have ever thought about in the context of epilepsy, if they might actually have anti-epileptic properties. Long story short, we looked through thousands of compounds and made a short list of candidates, and then we tested those candidates in a zebrafish assay to see if it could reduce or stop seizures, and we found a couple of good candidates. So we published a paper. We thought that was really cool, but that was kind of all we really thought about at that point. And then I was contacted by a woman, and I have her permission to share this because I think it's a very compelling story. But a French Canadian mom contacted us late last year. She had an 8-year-old son who had had intractable seizures since he was 2. They had tried everything. The neurologists were stumped. They couldn't figure out, how to stop these seizures. So another member of their family was diagnosed with roundworm, and the whole family began treatment with Parentel. Now, I didn't point this out, but Parentel was one of the compounds that we shortlisted in our study and that we found in zebrafish was effective at stopping seizures. So this is a drug that was developed for parasites. And to her astonishment, her 8-year-old seizures just stopped. I mean, he was having them every single day. And it was just a night and day difference when the whole family was taking this. And they had no seizures for 2 weeks, which was just unbelievable. The problem is she's had a real hard time getting this drug prescribed because nobody believes her, even though she showed them our paper and that there is objective evidence. Nobody, is believing her. So, long story short, this is a study that ultimately grew out of genetic research that found drugs that match a certain profile for epilepsy, and is kind of an individualized medicine approach. Let's see. So, yeah, this is kind of also out of context, but I probably actually shouldn't have put this one in here. Anyway, I'm a believer in this approach of using genetics for more individualized treatment prescription. One other thing I wanted to, this was from a grand rounds I gave a little while ago. I put a picture in there just to kind of help you visualize this is not who the person is, but the data are real, right? So we pulled this from one of our participants because I was specifically looking for, and I'm not showing you case 1 or 2, but I thought this was also a striking case. I wanted to identify people who had participated in our research and in the clinics had been diagnosed with one thing, and I am not casting doubt on that diagnosis at all, but I'm just kind of raising the possibility that genetic data might be useful for thinking more deeply about what is going on. So he was a 10-year-old male, presented with hyperactivity and a variety of symptoms that were perfectly consistent with ADHD. He also had an expressive language disorder. We looked at his polygenic scores and for major psychiatric conditions, And for the polygenic score for ADHD, he was totally in the normal range. There was no high levels of ADHD risk, no high levels for autism risk. He was actually found to be resilient against major depression. He was only in the 13th percentile for risk for depression, which means he's on the protected side. But he was in the 99th percentile for risk for bipolar 1. Now, at 10 years old, that is well before the age when most cases of bipolar would be diagnosed. And I think probably in most child psychiatry clinics, you're probably not thinking about bipolar disorder as your first line diagnosis. But I've talked to a lot of psychiatrists about this, and I think, this knowledge of where the genetic risk actually is would give you pause a little bit, both about prescribing a stimulant, but also kind of following the trajectory of this person as they approach young adulthood and get more in the time when something like bipolar 1 and a psychotic break would would happen for the first time. Um, so that's another example of where genetics shows promise for at least refining the diagnoses that we are giving in our clinic. All right, I'm taking note of the time now. We have, by my watch, we've got about 15 minutes left. I'll say just a few things about a study we've done on suicide risk in autism, and then hopefully we'll have plenty of time for questions afterwards. So this is your cue to start thinking about some questions to put in the chat so we can have a good discussion. Just a few definitions, and I want to emphasize the 988 hotline. If you or a loved one are in crisis, please know that there is a helpline here. That's 988. The getting the terminology right is important when we talk about this. So suicidal ideation or suicidal thoughts are reported thoughts of engaging in suicide-related behavior. An attempted suicide is a non-fatal self-directed act intended to result in death. Completed suicide is a self-directed act with the intention to die that results in that person's death, also death by suicide. Suicidality, broader spectrum of thoughts, behaviors that encompasses suicidal ideation, attempts, and completion. And then suicide survivors are family members, significant others, or acquaintances who have experienced the loss of a loved one due to suicide. Oh, shoot. Well, formatting got messed up when I carried this one over. Sorry about that. These are a few excerpts. So this paper, unbeknownst to us, when it was published early last year, was picked up on the Reddit science community, and there was a very vigorous discussion with over 600 comments commenting on the paper that I'll talk about in just a minute. But I wanted to have some in their own words, and these are largely self-identifying autistic people talking about how this resonates with them. So when you don't fit into society and are smart enough to know just how stacked it all is against you, you start looking for an exit. You feel incompatible with the world, like there's something fundamentally wrong with either you or the world. Like nothing you can do will ever change that fundamental rejection. And since you can't change reality, the only option available is then to remove yourself from it. And I can see, dang, yeah, I'm not sure how the formatting got screwed up. Apologize for that. This is on a more positive note, a person that recounted struggling with suicidality in adolescence and then coming through into adulthood and the things that were helpful to him. So this person said that it helps me be self-assured that my interests and routines and all of that are okay. And I shouldn't beat myself up over them, even if other people don't understand. If you can help him, he's responding to another person talking about their son. If you can help him cultivate this in some way, I think it will go very far to help. The second biggest thing is accepting that I have autism. And with that, I have additional care needs and limits that I must listen to. I've started to learn to set limits for myself and to say no, to not keep pushing my social battery when I'm all worn out. That person goes on, unfortunately, it's chopped off, but the person goes on to say, if I had done 5% less, I would have felt 50% better. So that's kind of a profile that you'll see when I tell you about the results of the paper. This is just a little bit of background. of the statistics. So in terms of death by suicide, autistic individuals have a almost 8-fold increase in risk for dying by suicide than non-autistic people, and are 3 times as likely to attempt suicide. There have been some studies linking the genetics. There, in terms of non-genetic risk factors, there's lack of social support, depressive symptoms, and the intensity of the autistic symptoms have been found to be risk factors. And then protective factors. One thing they found is that in other outside of autism, things that are normally protective factors like education or IQ or age or marriage, those may not extend their effects to their beneficial effects to autistic individuals. And that's something I think that is key for clinicians to understand. This is from our paper. It's also very important to know that IQ delays diagnosis. Okay. So this is parent reported IQ, which is very fuzzy. They put it into brackets, and then we have IQ from clinical records, and they both say the same thing, which basically we broke up the lines of this is age at diagnosis for autism. Orange line is if you have an IQ of 120 or greater, and then the blue line is if you have intellectual disability. So you can see that you're pushed to the right in terms of age at diagnosis, the higher your IQ is. And what that translates to is fewer years with the support that you need and the recognition that you have a neurodevelopmental disorder. So we worked with the Bell and Blank Center here at the University of Iowa, and we also combined data from Spark and another study called ABCD, various kinds of data. And basically what we found was evidence that individuals or that higher cognitive ability is a risk factor specifically among autistic people. So autistic children had more suicidal thoughts than non-autistic children, about 14%. Autistic children with suicidal thoughts had higher IQ than those without. Autism diagnosis and cognitive ability show evidence of an interaction tied to suicidal thoughts as an outcome. Basically, higher cognitive ability is protective in non-autistic individuals, but it is a risk factor in autism. Okay, that's another clinically relevant thing to recognize. If you have a very bright autistic individual, then you will want to have that on your mind as you are helping them. In the interest of time, we'll kind of scoot through this a little bit. These are a little more technical. This is also a little bit more technical. If it comes up in discussion, we can talk more about it. I'm just about at the end here. Okay, so to sum up, yeah, again, the higher cognitive ability was a specific risk factor among autistic people. It's normally protective in non-autistic people. We also found that higher polygenic scores for cognitive performance are associated with endorsement of suicidal thoughts and Spark. And we think that that also extends to undiagnosed parents. So parents with higher educational attainment polygenic scores were more likely to endorse thoughts of suicide for themselves. And these are just some figures showing that. So the conclusions of this are that high IQ delays diagnosis and the provision of services and support. And compared to people who are not diagnosed, high IQ increases the risk of suicidal ideation, and polygenic scores for those things index higher suicidality risk in both children and parents. That's the end of the talk, and I hope that we have time for a couple questions here.
Great. Thank you, Dr. Michaelson. We do have some time to take questions, so please type your questions in the Q&A box. We do have a couple, but I want to mention our continuing education real quick. So I am putting that link to the course evaluation where you can print your certificate of attendance in the chat box right now. And I will also be sending out an email with the recording and with step-by-step instructions on how to claim the continuing education credits through the Carver College of Medicine online CME portal. Okay, so a couple questions that came in. I have a 5-year-old non-speaking autistic daughter. Is it worth it to do genetic testing? What might we find out?
I would say if you're well, find out if your insurance covers it. If it covers it, then absolutely do it because it can, it may be that you don't find anything with the genetic testing, but as I mentioned before, if there is a specific syndrome that it might be because of the genetic result, then you'll have a more specific community of people who are dealing with something that is more that more closely resembles your experience. And so I think that kind of improves the quality of the support. You also might find that you're eligible for certain clinical trials if that's something that you're interested in. So I think I I can't imagine that especially since there's already a diagnosis, there's not a lot of harm that can come from getting the genetic facts that might pertain to that as well. I see the second question is,
Yeah.
did the diagnosis change like with case three? That is such an interesting question. And right now, the answer to that is there are no protocols for this. There's the DSM is going to, I hope in the next version or maybe the one after that, there's going to be at a ground level how to integrate genetic results in the diagnostic process because the DSM has all sorts of information about differential diagnosis. What it doesn't have is solid guidance on the impending genetic stuff that people are gonna have to start to reckon with. And so the people I talked to said, and my impression, I'm not a clinician, but I would say that this child, it was not an error that they met the criteria for ADHD, but it certainly complicates the picture when you think, well, okay, they do meet diagnostic criteria, but the biology is telling me that this is probably more likely prodromal bipolar type manifestation. And we should think about the long-term picture with that in mind. And we should make sure that we don't prescribe anything that might have a tendency to trigger a manic episode. Um, the next question about this is such a good question. How do you share suicide stats with parents without scaring them? And do you have any resources that could be shared to help mitigate the risk? I wish I had more to offer. We're working on that in terms of the resources, but I would just say this is a particular flavor of suicide risk that seems to be a little bit different than some others that we see in mental health. In that it catches a lot of parents. I've talked to a lot of parents in the wake of this paper coming out. I can't tell you how many parents, suicide survivors I've had email me in the wake of this paper and share their tragic stories. And one common thread that seems to go through a lot of them, not all, is that it came entirely by surprise. It was very well planned. It was very decisive, and it kind of came out of nowhere. And so I think parents should be proactive, especially okay, so here's the thing that is going to be different. Uh, if your child has high IQ and has certain areas where they're a very good performer, I think as a parent, it's very easy to think, oh, they already get a lot of positive reinforcement for their abilities. And they probably already have a really good sense of self-worth. You should not assume that that is the case just because your child is a good performer, but you should be very proactive about making sure that they have a sense of self-worth that comes from within. That's going to be something very important to attend to. Question about, I think it's important for all ID to have genetic testing. My son presented as autistic, but was diagnosed with Angel. That's a great example, perfect example. So that changes things. That's going to change that's going to change, that has care ramifications, that has all sorts of other community level, just the way you understand what your son is dealing with. So that's a great example of how genetic testing can kind of change the narrative a little bit. Is it false that male age affects risk of autism in their children? That's a really interesting question. We've done research on that, and I can briefly tell you that yes, it is true that the older a father is, because when we're talking about the the germline in the father, the sperm cells are constantly turning over. So you're accumulating mutations every time those cells are duplicated. So fathers accumulate more mutations in their germline. While that's been showed compellingly, what's less certain is that some uh, can we draw a specific connection to autism? We can draw a specific connection to more mutations, and that is indirectly going to increase risk for neurodevelopmental problems the older the father is. But it's hard to draw a straight line to autism. And then the last question, what about genetic disorders that present like ASD or result in the addition of ASD? Is that captured in genetic testing as well? Um, so if I think I understand the question, yes, that would be another scenario. So this is actually something really interesting and will be the final thing that I say because I know we're basically out of time. Um, age at onset, age at diagnosis. The if you are diagnosed in adulthood with autism, the probability of you having genetic risk that is for autism goes down the older you are. The genetic risk for other conditions, even if your presentation you feel like is consistent with autism, and I am not undermining anybody's self-identification here, but it is, we've looked at it in our data and we see that the older you are, the less likely the genetic cause is what we understand to be autism, and the more likely it is to be things like mood disorders and other neuropsychiatric conditions. So I think there's lots more research to be done there, but I think this question kind of gets at uh, what is autism, and I think that's something that the research field and clinicians are grappling with, and the community. Um, so I tried to answer as many as I could. I know we probably didn't get to everything, but and
No,
I think
thank you
we're out of
for
time.
your time and presentation
Yeah.
today, Dr. Michaelson. That will conclude our presentation, and I just want to sincerely thank you for the presentation and for all who attended. So have a wonderful afternoon.
Thanks, everybody. Bye.